In this short seminar, I will give an overview of the Entropy Variability website that is associated with the article that probably brought you here. It all started almost 20 years ago when Gert Oliveira came with the idea to describe the variability pattern that you find in columns in multiple sequence alignments by two parameters. One, the Shannon entropy, and the other one, the simple variability. So here to the left you see the top left corner of a very big multiple sequence alignment, and then for each column we determine the sequence entropy and the sequence variability to be explained in the next slides, and every column then becomes one dot in the plot at the right hand side. You would say, well, if you use two measures of the same thing, you get more or less a straight line. And indeed, it looks a little bit like a straight line, but there is quite a bit of spread around this straight line, both in the entropy and in the variability direction. And that is where the signal, as I will show, will come from. The Shannon entropy is simply the pi log pi for the 20 residues I, residue types I, at position P in the multiple sequence alignment, and the variability is the number of different residues observed in that column, although anything observed less than 0.5% is not counted, to avoid that we accidentally get bad results when we accidentally align something that shouldn't be in there, or when there are sequencing errors. In this plot, Gert, after studying almost 1,000 articles related to 5 proteins that in those days were the best studied from the whole PDB, he realized that certain areas are associated with a certain function. The bottom left, what he called box 1,1, it's pink in this plot, that's normally active site residues. And then with the same variability but higher entropy, in blue, we find the box 1,2, and those tend to be around the box 1,1 residues. They tend to provide support for the active site. Now we go totally to the other side, the top right, the green ones. We could actually never find any mutation information for these ones, indicating that every time when a mutation there was made, it didn't do anything, it didn't get published. This box here with the orange ones, it's the box 2, 3, those tend to be residues involved in modulator binding. For a G protein coupled receptor, the active site is where the G protein binds, and where the ligand binds is the modulator. Or for other proteins, there are other modulator sites. Well, use your fantasy, but nuclear hormone receptors, for example, they have also a ligand binding site, that's the modulator. And a second modulator is the dimer interface, and there where the co-activator binds is the main active site. And then we have the gray residues here left, so they have lower entropy than the modulator, but higher variability than the ones around the active site. And these residues are in the structure generally found between the orange and the blue ones. And often they're misannotated in the older literature. Sometimes they are called part of the modulator, sometimes part of the active site, because these residues communicate between the modulator and the main active site. And therefore, if you mutate here, it depends on your assay whether you think it's part of this or part of this. So, Gert could not really go much further than this, and now 20 years later, with much bigger computers, better software, and a much better understanding of neural networks and deep learning, Daniel decided to look at this same problem again and see which would be the two best parameters to describe in just 2 dimensions the variability pattern. And he used some neural network, although I would personally call it just a trick with matrices, but strictly formally speaking, it's a neural network, and an enormous amount of data. The enormous amount of data is in the human genome HSSP files that Koos Baakman has made And he made 27,000 something multiple sequence alignments with a total of 7 million alignment columns. HSSP files were originally thought of by Reinhard Schneider already in the early 90s, and they were later used by Burkhard Ross as input for neural networks for secondary structure prediction, which actually was also the main reason that Reinhard made them in the first place, and this article is here. Neural networks, or matrix methods as I would call it in this case, require numerical input, not text input, so every column is replaced by a frequency factor for the 20 amino acids. So here if we take one column and we count what is in there, then you see Q 7 times 23%, or 0.23 is the frequency. So this is the frequency per amino acid, but we are not interested in amino acids, we're interested in the variability patterns. So we use the sorted variability factor. So the highest frequency, whichever residue that is number 1, the second highest number 2, etc. And then comes the method. We start with a factor of length 20. We reduce that to 15, and that should be the best factor of length 15 to go back later from 15 to 20 again. And then from 15 we go to 10 to 5 and to 2. Here you see it worked out in greater detail. The idea is you have this factor, you multiply with a 20 by 15 matrix, then you multiply with a 15 by 10 by 5, 5 by 2, and then we go back 2 by 5, 5 by 10, 10 by 15, 15 by 20, and then hopefully we end up at the same factor. Of course we don't, there will always be differences. That's how life works. So we start with this factor, we go 20 to 15 and 15 to 20 and hope to get back at the same factor. So first we optimize these two, 20 by 15 and 15 by 20. As somebody who works a lot with symmetry, I would have loved this matrix to be the inverse of that one, but people who understand these things better than I do explained that it would be better to have two individual matrices and optimize both of them so that you get the best result. And on the website we describe a little bit more how these things are done and whose ideas and software are used. But the idea is to go from 20 to 15 and back to 20, and that for 7 million factors with a total minimal error. And so then we do 15, 10 and 10, 15, etc. And finally 5 times 2 and 2 times 5. And that is the whole thing. At the end, once we have the whole system set up, there are a few rounds of total optimization where everything at the same time can change. But okay, that's how the method works. And you can read that in the articles describing the method that we used. This is not about matrix methods, it's about variability factors. So that's the method. Now the results. What did the neural network, the bunch of matrices, what did it learn? Well, here we have the entropy variability plot, and when we put all the points not in entropy variability space, but in neuron 1 and neuron 2, so the two-dimensional space of the factor of length 2 that we got in the middle of our method, then you see that here is box 1, 1, here we get box 1, 2, 2, 2, 2, 3, and 3, 3. So these here the colors are nicely separated, pink, blue, gray, orange, green, and here it is pink, blue, gray, orange, green. So it looks as if entropy and variability, just with different separators, is coming nicely back from this method that didn't know what it was doing. This is just totally unsupervised. When we then color by entropy, you do see that it goes from red to green quite nicely. There's a bit of mixing here and here, but you quite nicely see that entropy goes in one direction. And then when we color by variability, it doesn't go in the orthogonal direction, but you see here variability 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It really goes quite nicely with a bit of mess when we get at the really high entropy, where things are so variable that, well, God knows what happens. It can even be misalignment in this region. Yeah, but you see that variability is really coming back in this method. So here you see the summary, and indeed when we take 27,000 multiple sequence alignments for all the human proteins found in freeze 19 of the human genome, then we do find that once we represent the variability columns in 2 dimensions, entropy and variability are those 2 dimensions. Okay, now having done this for entropy variability, looking at variability factors, what now if we don't sort the variability factors? So we're actually looking at the amino acids. Yeah, so we take again this same column, and now if the count is this and the frequencies are this, then we do not sort, but we use this factor 7 million times. We do the whole process again, and then you see certain factors come in and something somewhat similar comes out. It's not always beautiful, but if we now take a look at the 2 neurons, and for each combination of 2 we ask, what is the single residue that comes back perfectly, then we find in this 2-dimensional space, we find the residues back. Not only do we find the residues back, we find them back in a sensible way. We find residues next to each other that if we were looking at a multiple sequence alignment, we would say, yeah, they belong in the same column. Or if we were doing protein engineering, we would say, yes, that's a useful mutation to make. Obviously, it can't be perfect. Tryptophan is sitting next to glycine. That's not what a protein engineer would really expect, but then we will find quite a bit more beauty in here. For example, the hydrophobic things are to the left, the hydrophilic ones, the charged ones to the right. Histidine is sometimes charged, the polar ones here. And then we see serine really close as threonine a bit further, and threonine has to be here because it has to be close to valine, and because valine to threonine or alanine to serine are similar, or phenylalanine to tyrosine, similar in that it adds an oxygen. For example, you find many more of these things in here. Yeah, the positive and negative. Negative here. This is positive, negative, neutral. Positive here. The polar ones, yeah, we find them here in one group connected in the middle. The special cases, glycine, proline, cysteine, next to each other here. Yeah, so neural representations often also describe semantic relations. For example, if you go from asparagine to aspartic acid or from glutamine to glutamic acid, chemically it's the same difference. And in the 2-dimensional plot, it's the same vector. Similarly, valine to threonine, alanine to serine, phenylalanine to tyrosine adds an oxygen. Although here it doesn't add an oxygen, it replaces a carbon for an oxygen, and consequently the distance between these two is a bit bigger than the distance between these two and these two. From asparagine to glutamine and from aspartate to glutamate, in both cases you add 1 carbon and you get the same vector in the plot. So, what do we conclude? Well, with this very simple matrix approach, which formally speaking you can call a neural network, we can learn in a totally unsupervised manner what is going on in variability space if either we only look at variability or if we include the amino acid types underlying it.